Hi, my name is Susan Olalekan. I'm a graduate student at Rush University Medical Center.
Hi, my name is Hadijat Makinde. I'm a graduate student at the Immunology department of the Rush University Medical Center. I work with Dr. Greg Spear and I study the innate immune response of the lower female genital tract. Specifically, I'm looking at the role of IL-22 and IL-17 and the induction of antimicrobial peptides in response to sexually transmitted infections.
So what do I expect to learn from this conference? I expect to learn new approaches of studying Tregs, more importantly, how to identify the different subset of Tregs and their role in autoimmunity.
What am I learning? What do I hope to learn from this conference? I hope to learn more about the innate immune responses. Also, I hope to learn more about antimicrobial peptides and how they can contribute to fighting infection, especially in a human model system. Today is really the first official day, so I haven't had the chance to see a lot of speakers yet, but I did see a speaker this morning who talked about SPUNK1, which is actually a gene that is involved in a lot of antimicrobial regulation activities, and it was really interesting to me because he addressed two of the antimicrobial peptides that I'm actually looking at, which is cathelicidin and beta defensins and lactoferrin, so actually three. And he actually showed what adverse effect deleting that actual gene could have on induction of those antimicrobial peptides, and I thought that was really interesting.
So I saw a speaker this morning who spoke about B-cell and T-cell interaction in autoimmunity. I was very interested in that talk. I didn't really get what I wanted because he focused on B-cell as cytokine producers instead of B-cells as antibody producers. And he talked about new cytokines that B-cells are producing and their role in multiple sclerosis.
There are a lot of important things about immunology, so it's going to be hard to pin down one. But I think for me, the most important thing about immunology is trying to figure out how everything comes together as part of a whole. At least for me. So we're trying to figure out how T cells influence B cells, how B cells influence T cells, how they'll influence innate immunity, how everything influences one another, how the little parts come together as a whole, and I think that's the most important thing about immunology.
The most important thing about immunology is that it affects everything, and everything affects it, both intrinsic and extrinsic factors. What I love most about what I'm doing right now is when I think about something or I have an idea and I test it out and it works, or like when I attend a conference like this and I get an idea or see something that someone did and I try it and it makes my work, I progress with what I'm doing. For instance, I listened to Dr. Mac Jenkins once, and he works with tetramers, and I was beginning to do work with tetramers, but I never could pull out the population. So I saw that he used CD44 in his plotting gate, and I used that, and it pulled out my population beautifully. And ever since then, I always label my tetramers, do my tetramer staining with CD44, and I get beautiful data. So I love that.
What do I love most about what I do? What makes me excited? Well, I get excited about a lot of things, especially when it comes to my work. And she thinks I'm a geek, but I'm not. Just like she said, you do experiments or you do incubation, research, and then it doesn't work out. You do it again, it doesn't work out, and then you do it the 10th time and it works out. And I literally dance. I jump and I dance. Or I work with human samples, and a lot of it involves tissue work, which is not easy. And when I eventually get a nice single cell isolation from chunks of cervical tissue, and I'm able to view it under the microscope and actually see the different subsets, like the T cells or the platelets or even the blood cells, and I actually see epithelial cells, and I can tell all of this cell subsets differently. I go call my PI and I'm dancing, like, come and look at it, it's really nice. And that makes me happy, because I know that I've put in a lot of work and I'm actually seeing the fruit of my work, and it makes me happy.